Eukaryotic gene expression. Eukaryotic gene expression can be in the form of regulated at different stages. It can be regulated at the level of transcription, at the level of RNA processing, at the level of translation of the proteins, and at the level of post-translation. So in case of eukaryotes, the gene expression can be controlled at different points. It can be controlled at transcription level, at RNA processing level, at the translational level, as well as at the post-translational level. In this diagram, we can see that in the nucleus, this is the nucleus, in which DNA is producing RNA and RNA is producing messenger RNA. So, in case of eukaryotic gene expression, it can be controlled at the level of transcription when DNA is being converted into the RNA. And it can also be controlled at the level of when RNA is being converted into messenger RNA. And messenger RNA when it is going to produce the polypeptides. So gene regulation can be at the level of transcription, it can be at the level of RNA processing, it can be at the level of translation, even at the level of post-translation. Both intracellular signaling and intercellular communication are important for transcriptional regulation in case of eukaryotes. In case of eukaryotes, there are transcription factors that positive and negative control proteins. There are general transcription factors and there are specific transcription factors. They actually control either in the form of positive regulator proteins or in the form of negative regulator proteins. Transcription factors actually they bind to specific regions on the DNA and stimulate or inhibit the transcription. There are two types of the transcription factors. Transcription factors can be basal, or in other words, they are also called as general transcription factors, and specific transcription factors. Basal or general transcription factors are proteins that bind to specific DNA sequences within the promoter region to facilitate the RNA polymerase binding with that region. While on the other hand, specific transcription factors are those factors that are the proteins, they are actually the specific proteins that bind to response elements or to sequences which are called as enhancers or silencers, which are located near to gene and facilitate the action of BTF. In other words, basic transcription factors. In this diagram, we can see that how the DNA sequences control the transcription. This is the structure of the gene. Here is the Tata box, and this region is called as core promoter region. And this is the transcribed region. So RNA polymerase attaches to the DNA in the core promoter region, and in the same vicinity there is a Tata box, that is a specific DNA sequence close to the gene, and there is a binding of the transcription factors. So these are transcription factors or the basal or general transcription factors that bind to this region. So all these general transcription factors, they control the transcription of the eukaryotic genes. Now what are the enhancers? Enhancers act over relatively from large distance. The effects of enhancers are independent of the position where they are located on the gene. They may be upstream, they may be downstream, and they can also be within the genes. So enhancers, promoter sequences is actually those sequences which are close to the gene. While on the other hand, enhancers are those sequences that can be relatively far from the genes. They can be towards the 5' end of the genes, they can be towards the 3' end of the genes, and they can even also present within the genes. So those sequences that enhance the function of the gene or expression of the genes, they are called as enhancers. In this diagram, we can see that what are control elements, what are enhancers and silencers. So these are all control. This is the structure of a eukaryotic gene. These are the axon 1, axon 2, and within the axon there are introns. This is 5 translated region. Here is the 3 translated region. So this is the promoter region where there is Tata box. There can be Tata box. There are cat box. There is GC box. So the location of the Tata box, cat box, and GC box has been mentioned here and has been shown here. So these are the sequences which are called as enhancers and silencers, which are actually far away from the promoter sequences, and they can be towards the 5 end, they can be towards the 3 end of the gene, and they can even present within the sequence of the gene. So all these elements, they control the gene expression of the eukaryotes. Here we can see that Tata box is here and promoter sequence is here. This is the gene that have to be transcribed, and enhancer sequence is here. In this diagram, we can also see enhancers and journal transcription factors. These are all the journal transcription factors which actually facilitate the binding of the RNA polymerase with the gene region or with the promoter region. So here are the enhancers. This is the sequence of the DNA. Here is the enhancer. There is another enhancer here and other enhancer here. So enhancers are actually those sequences. Due to the presence of enhancer, the expression of the eukaryotic genes increases. Post-transcriptional regulation. Mostly control of gene expression is at the level of transcription in case of eukaryotes. Gene expression can be controlled even after the transcription. That can be in the form of RNA interference, that can be in the form of RNA editing, alternative splicing, and messenger RNA degradation. So if we conclude that about the gene expression in case of eukaryotes, most of the time this expression is controlled at the level of transcription, but it can also be controlled at the level of RNA interference, editing of the messenger RNA, splicing and degradation of the messenger RNA. Even there are certain other factors that control the eukaryotic gene expression.